Good morning again. We are going to thank you for joining us in this online IBEC seminar. First of all, some practical information. Please make sure to switch off your cameras and mics. At the end of the presentation, you will have the opportunity to make questions. Please write them in the chat, indicating if you don't have a mic. Now, let me introduce you our speaker today, Xavier Trepat, Group Leader of the Integrative Cell and Tissue Dynamic Group of IBEC. You all know Xavier and his outstanding career, so I will be very brief. Xavier Trepat received a BSc in Physics in 2000 and a BSc in Engineering in 2001. In 2004, he obtained his PhD from the medical school at the University of Barcelona. He then joined the program in Molecular and Integrative Physiological Sciences at Harvard University as a postdoc. In 2008, he became a Ramon y Cajal researcher at the University of Barcelona, and in 2011, an ICREA research professor at IBEC. In 2015, he won the Bank of Sabadell Award for Biomedical Research, and in 2018, he was elected EMBO member. He has been awarded with 4 grants from the European Research Council, the last one an ERC Advanced Grant to develop the EpiFold project, focused on understanding the principles and mechanical properties of 3D epithelium. Xavier, thank you for being here today. The floor is yours.
Thank you very much, Teresa, for the kind introduction, and thanks everyone for showing up this morning. And it is a pleasure to be here and to be able to tell you new research in our lab, and specifically our recent projects on the mechanobiology of intestinal organoids. My lab is generally interested in epithelial tissues, which, as you know, are the thin barriers that line the internal and external cavities of your body. Epithelia can come in many different shapes and forms, but the simplest of these forms is just one layer of cells that are closely packed. They are connected to different types of junctions, such as tight junctions, adherence junctions, and desmosomes, and they are usually adhered on top of a complex hydrogel that is called the extracellular matrix. Now, despite the fact that these very thin layers account for a very small fraction of the mass of your body, these tissues account for a number of functions that are core to physiology and to disease. For example, they are responsible for tissue compartmentalization. They are the first barrier in the protection against pathogens. They play a central role in morphogenesis, and in particular, they drive early morphogenetic movements. They secrete and transport ions, fluids, and biomolecules. They are central to wound healing and tissue regeneration, and unfortunately, they are also responsible for about 80% of all tumors that are known. Now, to perform all these functions in physiology and disease, epithelia must be able to perform a number of biological tasks, such as migration, such as division, differentiation, extrusion, folding, and many others. So I call these mechanical tasks because they are either directly driven by physical forces or they are heavily influenced by physical forces. For example, you cannot migrate without a force that propels you. You cannot divide if there's not a force that separates the daughter cells. You cannot be extruded if there's not a force that kicks you out of the monolayer. You cannot fold without a moment of a plane. And so functions that are traditionally associated with just biochemical regulations, such as differentiation, such as stiffness, we know today that they are also heavily influenced by mechanical forces. So that's why I call epithelia mechanical multitaskers, because they must be able to perform all these mechanical tasks, and they must be able to do that at the same time and at the same place. So the main theme of this talk would be to ask what are the principles that enable epithelia to behave as mechanical multitaskers. Now, as an introduction, I would like to go through what we've learned in the past decade or so about some of these mechanical tasks and how they are integrated. And I would like to begin with this example that that we studied some years ago in the lab. So this is a classical experiment in epithelial biology. It's a wound healing experiment. You start with a monolayer of epithelial cells, you wound it with a laser with whatever object, and then you just watch how cells close that wound. Despite the fact that this is an experiment that you could run 100 years ago, and in fact it was run 100 years ago, we still do not understand this fundamental problem. And when we started worrying about this, we soon realized that it was impossible to be able to understand this problem without having access to the physical forces that these cells exert on their surrounding in order to close this gap. So for that reason, we've spent a lot of efforts over the past decades in order to develop technologies that would make these forces visible. And in this problem, there are two main relevant forces. One is the forces that are called traction forces. These are the forces at the interface between a cell and its underlying substrate, which we measure with a technology that is called traction force microscopy. I will not give you the details of this technology, but essentially we sit cells on top of a very soft substrate. And from the deformations of this substrate, then we can infer what is the force that cells are exerting on the substrate on a pixel by pixel basis. Another type of relevant forces in this problem is cell forces, which we measure with a technology that is called monolayer stress microscopy. Now, these two types of forces are not fully independent. In fact, depending on the geometry, they are fully dependent. For example, in this example of a tug of war, these guys are generating a force on the substrate, this traction force, and this force is sufficient to determine the tension within this rope. So, of course, in 2 dimensions, the problem is more complicated, but this sort of gives you an intuition that these two types of forces are not fully independent. Of course, there's many other laboratories that have been developing technologies to measure forces. And in fact, we just wrote a review about exactly this subject, measuring mechanical stress in living tissues, that I invite you to read if you're interested in this type of topic. Okay, so with these technologies in hand, now we can turn a movie that originally looks like this into one that looks like this, in which we now have a map of the forces that cells are exerting at every point of space and at any instant of time. So these are traction forces, forces that cells exert on their substrate. And notice that they are color-coded according to the sign of the radial dimension. So forces that are pointing away from the wound are red, forces that point towards the wound are green. And if you watch this movie carefully, you'll notice that there is a transition between forces that are predominantly red at the beginning of the experiment to forces that are predominantly green at the end of the experiment. And this corresponds to the different mechanisms of wound closure. Red forces are associated with cell migration, cells grab on the substrates and propel the body forward. Whereas green forces are associated with the assembly of a supracellular cable lining the edge of the wound, and the contraction of this cable closes the wound in a mechanism that's analogous to a purse string. So once you have these force maps and the dynamics of the full dynamics of the system, then you can start writing models like this one that we did with Boeing Broadland and Vito Conte, in which we can write a vertex model where we can input cell migration through these red lamellipodia as well as a contractile ring. So then going back and forth between experiment theory, then we can nail which are the mechanisms that drive wound closure. Now this has been published a while ago, but I wanted to begin with this movie because I think it illustrates nicely how there are some fundamental problems in biology that you can only address when you can actually measure the forces that cells are exerting. And once you measure those forces, then you can fully understand mechanisms. Of course, I'm not claiming that we fully understand this one, but we think that we are much closer than when these forces were invisible. Okay, so the complementary problem to the closure of a wound is the expansion of a monolayer. And that's what you're seeing in these movies here. So this is a monolayer of breast epithelial cells that are expanding from an initial micropattern. Now overlaid on top of this monolayer, you can see the maps of cell-cell stresses. So we're no longer seeing attractions here. Now we're looking at forces that cells exert on their neighbors. Each panel in this movie corresponds to a knockdown of one protein of the cell-cell adhesome. By knocking down one at a time, the proteins of tight junctions, adenosine junctions, desmosomes, and cap junctions, we were able to infer what is their mechanical role. We were able to confirm some existing knowledge, and we were able to unveil some new mechanisms. Now, these two cases are cases of migration in which we're not superimposing any external signal. So the only signal that makes these cells migrate is the presence of a free edge, the geometric constraint. But of course, one can also superimpose gradients of chemical concentration or physical properties. And this is the experiment that Raymond Sounier ran in the lab some time ago, where he repeated this experiment, but rather than letting these cells expanding on just a flat uniform substrate, he seated these clusters on top of a gradient of stiffness. So one edge of the monolayer is seeing a soft edge that's going softer, and the other end is seeing a stiff edge that's going stiffer. And as you can surely appreciate, cells prefer to move towards stiff rather than towards soft. This is a phenomenon that is called eurotaxis, the tendency of cells to migrate towards stiffer regions. It had been discovered and studied for single cells, but what Raymond was able to show in epithelial cells is that when these cells are isolated, they are completely unable to eurotax. By contrast, when you put them together, then they develop an integrative mode of migration that allows them to migrate. So with some sort of collective intelligence, as it's called in the field. So Raymond was able to nail down the mechanism, which is the transmission of force from one edge to the other that enables this cluster to behave as a supracell. Okay, so these are three examples of migration in which cells are migrating on a flat substrate. But most of our tissues are not flat. So there's an additional component to this multitasking, which is on the first hand studying this in 3 dimensions, and the second one is to integrate this migration with other functions, such as, for example, cell division. So let me start with the second point, which is how can one integrate mechanically cell migration and cell division. So for that, Marina Oroz, a former PhD student in the lab, she expressed these Fucci constructs in MDCK epithelial cells. So for those of you who are unfamiliar with Fucci, Fucci is a system that labels the nucleus of your cells with a different color depending on their state in the cell cycle. So during G1, the nuclei are red, then they turn yellow, and during S, G2, and M, they are green, and then they become colorless. So with this tool, we can now monitor the position of every nucleus. We can monitor their velocity, their distance, their size, and in addition, we can monitor their state in the phase cycle. So Marina measured all these variables. She also measured the tractions that these cells are exerting as they expand. And she was also able to measure the tension for cells that exert on each other as they expand. With all this arsenal of variables that she was able to very carefully measure, then she was able to ask whether there's one of these mechanical properties that would be predictive for the duration of the cell cycle. And what she found was that there is one particular variable that was highly predictive, and this variable was the tension that cells are exerting at the duration of the movie. To show, if you know the state of tension, the tension that one cell is subjected to during the first few hours of the cycle, then you can predict the duration of the first phase of the cycle, G1, but also the duration of S, G2, and M. So what these data show is that the forces that we had traditionally being associating to cell migration are also governing the duration of the cell cycle. An example that illustrates how migration and division can be integrated through mechanical forces. So the third layer of integration, functional integration that I wanted to point out is folding. So as I said, most of our tissues are folded. So when cells perform their function, they must perform it. At the same time, they are sustaining or even driving folding. And this is illustrated in this classical movie of gastrulation in Drosophila. You start with an ellipsoidal epithelium, and eventually there's invagination of the ventral furrow, which is governed by a bending mechanism. So there is an accumulation of myosin, which you see here fluorescently labeled, and this accumulation of myosin causes a contraction that then drives the invagination of this tissue. So this is an example of folding governed by bending. There are other mechanisms that allow epithelial tissues to bend. This one is from Guillaume Charas's lab. where they were able to grow a monolayer between two cantilevers and then bring together the two cantilevers. And when they do that, they were able to observe that the monolayer is buckling. So this is buckling driven by the applications of an exogenous force, but buckling can also proceed through growth. And this was shown here in this paper by Aurélien Roux's lab, where they were able to generate capsules, which you see here in blue, and to grow epithelia inside these capsules. So when epithelia reach confluency, eventually they build up a compressive stress. And at some point, this compressive stress is so large that it causes the delamination of one part of the monolayer, which you can see here. So this is a similar example here, except that here it's driven by cell growth, which is another example in which it shows this multitasking, in which, in this case, growth and folding are completely coupled. And finally, there's another example that we studied in this paper led by Ernest Latorre in the lab. As you know, epithelia are extraordinary at transporting ions across their surfaces. And by transporting ions, you can create differences in osmotic pressure that can then drive flows. And that's exactly another mechanism for folding that we studied here in detail. Okay. So in this introduction, I told you about three functions that are folding, migration, and division. I showed you how they can be mechanically integrated. So to study this a bit further, we turn to a tissue that is probably the most proficient multitasker in our body, and this is the intestinal epithelium. This is a work that was driven by two PhD students, Carlos Pérez González and Gerardo Ceada, in collaboration with Daniela Vinovich at Institut Curie and Marino Arroyo on the theory site. We have uploaded this in the bioarchive, so everything that I will be telling you today from now on, you can read it in the bioarchive. So intestinal epithelium is shaped with these finger-like protrusions. And the purpose of these protrusions that are called villi is to pack a very large surface into very small volume in order to maximize intake of nutrients and fluids. Now, one remarkable feature of the intestinal epithelium is that it has the record of being the fastest self-renewing tissue in our body. So every 3 to 5 days, the entire surface of your intestine is renewed. So every cell that is currently lining the surface of your intestine will be gone in a week. The way this process works is through the action of stem cells that are located at the bottom of some regions that are called the crypts, which are located between the villi. So these are these crypts. And at the bottom of these crypts, there are two main cell types, stem cells and Paneth cells. So Paneth cells are secretory cells that I will not be telling you about. And stem cells are the ones that divide. Once they divide, they move to this region that is called the transit amplifying zone. Here they divide further, and then they move towards this villus surface where they differentiate and they migrate. At some point, they reach the tip of the villus where they are extruded. So during this journey from the bottom of the crypt to the tip of the villus, intestinal epithelial cells must be able to perform a number of core functions in biology. These functions are stemness, compartmentalization, folding, proliferation, migration, differentiation, and death. Now, these are not anecdotal functions in biology, right? So these are the core functions in biology. They are entire conferences dedicated to each one of these functions. Right? So what we wanted to understand is how are all these functions, that each of them has a mechanical component, how are they integrated in the system, and whether mechanics plays a role in that integration. There is a question that you cannot access in vivo because the system is not accessible optically or mechanically. But thanks to the development of the field of organoids, this gave us the opportunity to interrogate some of these questions in vitro in the lab. So as you know today, we can take one single stem cell, and if we provided the right physicochemical environment, then this cell will form an organoid, which I'm showing here and schematically here. So in the simplest form of these organoids, this is just one monolayer of cells that is folded, and that contains domains that are called either crypt-like domains or villus-like domains. Now, these domains have the right curvature and contain the right cell types. And of course, an organoid is not the exact same thing as the intestinal epithelium in vivo. But it's been shown today that these main core functions, to some extent, they are recapitulated by organoids. So in order to understand how the system works in vivo, these models are outstanding to understand the problems in vitro. Still, to understand the mechanics of this problem, in 3D is problematic because there is no current technology in 3D that will allow you to compute how much force one cell is exerting on its neighbors and on its surrounding as a cellular matrix. So for that, we went one step further in simplification of the system, and we tried to build a new model of organoid, which is based on organoids that are cultured on soft 2D substrates. Now, the way that this model works is the following. So you begin with a 3D organoid, and we cut it into pieces. When you do that, more cells will die, but the cells at the stem cell niche will not die. They will adhere on the underlying soft substrate coated with collagen and laminin 1. And then after that, they will form a monolayer. This is illustrated in this movie here that shows you the first hours right after dropping these pieces of organoids on top of our soft and flat substrate. So this movie is showing you how cells migrate, how they organize, how they self-assemble, and finally, they end up forming a monolayer in which there's an area where you can say differentiated epithelium, and then there are areas that are sort of structurally more complex. Let me play that movie again so you can see how you start with a very disordered system, and eventually the system self-assembles so that it forms just a monolayer of tissue. So we focused on these regions that seem to have a more complicated geometry. When you take a zoom of those areas, they look like this. Now, this looks like a three-dimensional projection of a tissue that many of us are used to look at when we do confocal microscopy. But it turns out that this is just one single plane of confocal microscopy. This is just one confocal plane. And as you can tell by the different shape of the cells, here there's certainly different cell types. So the first thing that we did was to develop, to use actually the current arsenal of tools to identify which cell types you have in our system. So for example, we label stem cells, and what we found is that in the central area where cells are highly packed, this is where stem cells reside, labeled here with LGR5. We also found that when we labeled, when we sent for Paneth cells using lysozyme, we also identified that Paneth cells are localized in this region. So these circles that we're observing, these are stem cell niches that contain only Paneth cells and stem cells, much as the niche in vivo. Now, outside these niches, then we can start seeing markers of differentiation, in this case, cytokeratin 20. So these and other evidence that I don't have time to show you told us that this system that is extremely simple and grown into D, it retains all the main cell types of the intestinal epithelium and their segregation. So they are segregated into a niche and into a villus-like domain. And not only that, but this system also retains the dynamics of this process. This is in fact the first snapshot of a movie in which, if you pay close attention, you will be able to see that cells at the stem cell niche divide, and then they migrate away from the stem cell niche, and they reach this area where they are very spread, and eventually in this area they are extruded into the flowing medium in top of these organoids. So not only the main cell types and the zonation of those cell types is retained in the system, but also to a great extent the dynamics of the tissue is also retained. You can appreciate better this cell death here in this movie. I'm now taking a higher plane of this exact same movie, and then you can say all these roundy cells, these are cells that are extruded from this area into, in this case, the overlying fluid. I can show you other movies that are faster, so that you can better appreciate, for example, division, all these cells that round up spontaneously in the system. So these are cell divisions. You can also appreciate cells that round up and are extruded here in this villus-like domain, and you can also appreciate this beautiful migration of differentiated cells. So we call these organoids, we call them 2D crypts, which is an oxymoron because you cannot be 2D and a crypt. We think this oxymoron captures well the fact that this extremely simplified system in 2D does everything it was supposed to do in vivo. And to me, this was really surprising, and it still is, because it goes against the way I think about epithelial biology and the way I think about tissue engineering. I think the way I think about these problems is that if you want a tissue to do what it's supposed to do in vivo, you need to provide it with the right 3D environment, right? So you have to provide it with the right geometry, the right extracellular matrix, the right ligands, the right density of those ligands, the right porosity of those ligands, the right stiffness of those ligands, the right growth factors. Sometimes you need to provide stromal cells and so on and so forth. And we're doing none of that here. What we're doing here is simply we chop brutally these organoids and we throw them on top of a substrate, and then they self-organize in order to perform exactly what they are supposed to perform. So we're doing all the wrong things, but these organoids fix them and do exactly what they are supposed to do. So this really calls into question the way, at least I thought, about what I call the 4 Fs, which is the form, the function, the fate, and the force of a tissue. So we decided to use this model in order to revisit this relationship with these 4 F words. So the first thing that we did is to measure the forces that these tissues exert on their underlying substrate. So for that, Carlos and Gerardo worked with Manu Gomez in the lab in order to map the 3-dimensional tractions that these cells exert on their underlying substrate. So these are 3-dimensional tractions. So to plot them, one needs to be a bit creative. And the way that we decided to plot them is the following. So in these yellow vectors, these are the XY tractions, which are 0 at the center of the stem cell niche. Then they grow as we go away from the stem cell niche. They reach a maximum, and then they decay, and eventually they change their sign. The color coding accounts for the normal components of forces. So forces that are blue are pointing towards the substrate. Forces that are red are pointing away from the substrate. So we took advantage from the fact that the system has sort of polar symmetry. So we averaged these data in 360, and we averaged several crypts, and then we were able to plot the traction of the 2 main components of this problem. These are the tractions that are normal to the substrate and the tractions that are parallel to the substrate, and we were able to plot those tractions as a function of the distance to the crypt center. And what Carlos and Gerardo were able to show is that this sort of relatively complicated force pattern, and certainly non-monotonic force patterns, they coincided with the functional compartment. So there are mechanical compartments and there are functional compartments, and these compartments colocalize. For example, the normal traction is highest at the center, and then it decreases. When it crosses 0 traction, this is the boundary between the stem cell niche and the transit amplifying zone. Then this increases and it slowly decreases. If you look at the radial traction, it is 0 at the center, and then it reaches a minimum that coincides roughly with the interface between the stem cell niche and the transit amplifying zone. And then this peaks negatively, and it grows again, and when it crosses 0, then we are again in an area in which there is a transition between 2 domains, in this case, the transit amplifying zone and the Witters-like domain. So there's a colocalization between mechanical and functional compartments, which suggests that there is a mechanistic link between those. Of course, we can run these experiments as a function of time, and this is what you're showing here. And there's certainly fluctuations in the system, as you can see, especially in the Witters-like domain, but the main patterns are conserved. So this movie that lasts about 5 or 6 hours shows that there's a very clear force pattern that is sustained in time at the stem cell niche, at the transit amplifying zone, which is labeled here, marked here with these red forces, and even at the Witters-like domain. Perhaps a more intuitive way to look at this problem is to take a cut along this direction and look from the lateral side. And when you do that, you notice that the cells at the stem cell niche, which is here, are certainly pushing their underlying substrate, where cells at the transit amplifying zone, which are here and here, are pulling the substrate upwards. Now, so these non-monotonic traction patterns, they are complicated, but there are some features that are actually very simple. And sort of the first order thing that we sort of found or were able to think about in this problem was the fact that if the stem cell niche is pushing and the transit amplifying zone is pulling, then this creates a bending moment. So somehow what this monolayer is trying to do is to bend, right? If you take an inert material and you apply these forces, what you will do is you will bend it, right? However, here, this monolayer is not able to bend its substrate because the substrate is quite stiff. It's 5 kilopascal. But we then reason that if we make the substrate soft enough, then we should be able to see these monolayers bending. And that's indeed what we found. So we started with 15 kilopascals, in which the stem cell niche is bulging out of the monolayer. But as we were going to softer and softer substrates, and here, for example, 7.7 kilopascal, then the monolayer was progressively bending. And if you go to really, really soft substrates, like this 0.2 kilopascal, then the monolayer bends, and the crypt now adopts the morphology of a real crypt. So you really get bending to an extent that is similar to what you're observing in vivo. So we measured as well in each one of these systems the forces that cells are exerting, and we were able to find something that is quite remarkable. And this is that the normal force, so the force that is pushing the substrate, this force is independent of the substrate stiffness. So there's no mechanosensing here. So cells couldn't care less about the substrates. By contrast, the radial component, the radial that is parallel to the substrate, depends on rigidity. So there's no rigidity sensing in one component of the cytoskeleton of the forces, but there is clear rigidity sensing in the other one. So indicating that these different force components arise from different mechanisms, which we're now trying to understand. Now, all these data led us to ask the question of what is the mechanism that drives bending. So we know now that these monolayers are able to bend in the same way that they bend in 3D organoids and in vivo. Now the question is, what is the mechanism that allows them to bend? And as I said in my introduction, there are different mechanisms. The two that we think apply to this problem is one, myosin differential. So for example, you can have an accumulation of myosin on the apical surface of the cells that will create bending of the monolayer. And the other one is buckling. So for example, we know that cells at the niche are dividing heavily. So that can create a buildup of pressure that would eventually lead to buckling of the monolayer. These two mechanisms have been alluded to in the literature. And to test them, traction forces are not very useful because these two mechanisms lead to similar traction patterns. But there are some biochemical interventions that one can do in order to discern between these two mechanisms. Chiefly, if you inhibit myosin, this mechanism should go away, whereas this one should not. So we added blebistatin, a myosin 2 inhibitor, and this is what I'll show you in this movie. This is a movie that loops. We start with a baseline, which is here, then we add blebistatin here, and we wash away blebistatin. So again, baseline, blebistatin, and blebistatin is washed away. So you can probably appreciate that when we add blebistatin here, then all forces go away. When we wash out blebistatin, all forces reform. So myosin seems to be this knob that is sufficient to turn on and off in order to drive force generation in the system. And not only that, we were also able to show that when you add blebistatin, you inhibit myosin, and then the monolayers completely unfold. So folding is really driven by myosin. So suggesting that this buildup of pressure created by growth is not the mechanism at play here. So since it's myosin, one thing that one should do is to just visualize myosin and visualize actin. I'm showing you F-actin here. Myosin stainings look similar in terms of localization. So we know that there's an accumulation of myosin and actin at the apical surface of the stem cell compartment. This is something that was already known. But there's also an accumulation of myosin and actin at a boundary layer that forms a ring that surrounds this stem cell niche. You can see it better here. So notice that there's a number of cells that are organized in these stress fibers that seem to be contracting the niche that is inside. And so this again led to two potential mechanisms that are both dependent on myosin. The first one is apical constriction. So this myosin here contracts, and this creates bending of the monolayer. The other one is buckling caused by basal compression of transit amplifying cells. So these cells contract, and this creates bending of the cells that are inside, or buckling of the cells that are inside. Now, to discern between these two mechanisms, what one should do is to look at the sign of the stress in the system. In this case here, the mechanism of apical constriction, cells should be under tension. By contrast, in the case of basal compression, then cells at the niche should be under compression. Now, to know the sign of the stress tensor, so tension versus compression, what one must do is to cut the monolayer with a laser. And this is what Carlos did. So he took the monolayer and he cut it circumferentially at the interface between the transit amplifying zone and the villus-like domain. So he created a cut. I will play this movie. So he cuts along the red line. And as I'm sure you appreciated, once he did that, the monolayer recoils, right? So this part moves outwards and this part moves inwards. And here's a quantification of those velocity fields. He also cut at the boundary between the stem cell compartment and the transit amplifying zone. When he did that, he was also able to observe that there is recoil on both sides of the monolayer. And let me play this movie again. So there's recoil on both sides of the monolayer. We quantify these over many cells and monolayers. And what we found is that there's always a recoil. What this means is that the system is under tension. So the idea of buckling through compression is actually not applying to this system. So these and other evidence that I don't have time to go through led us to the conclusion that the stem cell compartment folds through apical constriction. So this was nice, but we wanted to go one step further. And what we wanted to do is to try to see if we could understand, just on basic mechanical terms, the shape of every cell in these organoids. We now know how the stem cell niche folds. We now want to understand the shape of every cell type in the system. So for that, we joined forces with Francisco Greco and Marino Arroyo at the UPC. So in Marino's group, they've developed a 3D Vertex model. And I will not have time to go through the details of this model, but essentially it's a monolayer of polyhedral cells that are attached to each other. And what this model can do very nicely is that you can prescribe a given tension to any surface of the cells. Then you can observe, you can ask, what is the consequence of a contraction of that surface of that cell? So we contract one cell or two cells or whatever surface in the system, and then we can observe how that impacts, for example, the shape of this monolayer. So what we did is so our first simplest approximation to this problem is, well, prescribe the tensions that will be proportional to the amounts of actin and myosin that we had measured experimentally. So we added a patch of contraction of high tension at the stem cell compartment, and the apical surface of the stem cell compartment. And we added a torus, a donut of tension that was on the apical surface, on the basal, on the basal surface of the cells in the transit amplifying zone. So Francisco and Marino then coupled this monolayer to a substrate so that we could actually measure the role of the substrate as well as the forces that cells are exerting. And what they did then is to come up with predictions that look like this, right? So this is one of the cells that we allow to, of the monolayers that we allow to relax. Then we can ask what is the shape of this monolayer as a consequence of the tensions that we prescribe. So Maria Matejic, a postdoc in the lab, took these results, these theoretical results, and she compared them with the actual experimental results. So she segmented carefully many crypts in 3D. And what she was able to show is that the model predictions were matching very closely our experiments. So cells were taller at the stem cell niche. They are tilted inwards as we're moving radially out. Then they adopt this banana shape that we also captured very nicely here. And then they relax toward this cuboidal shape. She was also able to do that on softer substrates, which again show this tilting towards the center of the monolayer, as well as this basal constriction and banana-like shape at the transit amplifying zone. So this was a very nice result because it showed us that by simply imposing the tensions that are proportional to the actin myosin amounts that we measure, then we can predict exactly the shape of every cell in this system. Of course, we did this much more carefully than I told you. So Maria did run a full morphometric analysis of these cells. I cannot tell you all that information, but feel free to look it up in our online manuscript. So I told you about shapes, but this model also captures the tractions, and in particular the normal traction that I'm showing here. So this is the prediction of the model, and this in orange here is the actual data. So notice that the tractions are fully captured in our model as well. Okay, so there is one question that I will not have time to go through, but there is a full section of our study. So far I told you a picture that is essentially static, but this system is highly dynamic, right? So that's what enables self-renewal. So a whole different set of questions in this system are what are the mechanisms that drive motions in this system that is self-renewing. So Carlos and Gerardo spent part of their spring confinement looking at these movies, segmenting cells in these movies, and then trying to visualize how cells, for example, wander around the stem cell niche, how they enter the transit amplifying zone, how they divide in the transit amplifying zone, how they migrate away from the niche in the villus-like domain. So they obtained many of these movies, they segmented many of these movies, and then they were able to ask, what is the mechanism that drives cellular motions? Now there's a clear hypothesis in the field, which is that cells are pushed. They are pushed by mitotic pressure. In other words, we know that cells are dividing heavily here. You can observe it live here. So this creates a pressure, and this pressure is the one that is pushing cells out of the crypt and moving them towards the villus-like domain. Now, our data are completely inconsistent with this idea because we know that the tissue is under tension all the time. And if the tissue is under tension, it means that no one is pushing anyone. In fact, what is happening is that there is tension. Unfortunately, I don't have time to go through all this evidence, but what Carlos and Gerardo were able to show is that these cells are dragged out of the stem cell niche through a gradient of tension. So the answer to this question, are cells pushed out of the niche by mitotic pressure? The answer is no. So this leads me to my conclusion slide. So I showed you that intestinal organoids exhibit a non-monotonic stress distribution that defines mechanical and functional compartments. We found that the stem cell compartment pushes the ECM and falls through apical constriction, whereas the transit amplifying zone pulls the ECM and elongates through basal constriction. I also show you that the shape and force distribution of the crypt can be largely explained by surface tensions following the measured apical and basal actomyosin density. I also showed you that cells are pulled out of the crypt along a gradient of increasing tension. In fact, this I didn't show you, but you can read it in our bioarchive preprint, rather than pushed by a compressive stress downstream of mitotic pressure. And finally, more in terms of an outlook, the mechanobiology of intestinal organoids is now amenable to experimental observation thanks to these models that allow us not to visualize every cell in these organoids, but also measure with single cell resolution or even with subcellular resolution the three-dimensional forces that cells are exerting in the system. So with that, I will just conclude, acknowledging the lab. I told you about the work of Gerardo and Carlos here. He was no longer in the lab when this picture was taken. Manu, they are surrounding Manu, who's sort of the brain behind many of the algorithms that I told you about, and Maria did all this careful segmentation of the organoids into the N3D. We had the collaboration of Marino Arroyo and his group, as well as Pera Roca-Kozaks and Natalia Castro, our technician, collaborations with Daniela Vinevich in Curie, and Edu Valle at the IRB. And of course, we're grateful to all our funders. So thank you for your attention, and I'll be happy to take any questions. Thank you very much.